So today is the last lecture for microbial genetics, and we are going to talk about basically a very intelligent uh, discovery in terms of uh, bacterial genetics, which led to discovery of gene regulation. And this is something which was discovered in bacteria, and many decades later, we learned that the fundamental principles of gene regulation which we discovered in bacteria or we learned from bacteria are also true in higher eukaryotes or in eukaryotes in other, so it's evolutionary conserved. So I'll just like to remind you about the central dogma. So normally DNA and the flow of information in a cell is from DNA to RNA to protein, so transcription of DNA and then translation of RNA into protein. In case of prokaryotes, there is no nucleus, no membrane-bound organelles. There is circular DNA which gets transcribed, and in bacteria you have what we call transcription and translation go almost together. So co-transcriptional or as well as co-transcription and co-translation uh, is going on. For example, this is DNA, RNA is being transcribed, and this RNA is caught up by ribosomes and they translate the protein. In eukaryotes, RNA gets transcribed, it undergoes processing, splicing, introns are removed, and then this mature messenger RNA is exported into cytoplasm where it is translated. Rest of the phenomenon in molecules, in organisms, be it a prokaryote or eukaryote, are same, which are the fundamental basis of heredity, which is basically, they replicate, they store information in DNA, and they are prone to mutation as well. Now, why we are looking at this central dogma or transcription and translation? Because today's topic about gene regulation, which was discovered in bacteria, is basically a process of transcription. So as I explained you, the process of transcription in prokaryotes and eukaryotes is very similar. The gene structure is you have promoter, you have the coding sequence, and then transcription terminator. In case of eukaryotes, you have promoter, the coding sequence is intervened by these introns, and then this pre-messenger RNA is transcribed. It undergoes splicing. The mature messenger RNA eventually is exported into the cytoplasm where it gets translated. Okay. So the transcription basically involves recognition of the promoter elements by transcription machinery, which is led by RNA polymerases. Now, why there is need to regulate gene expression? I used the word earlier, gene regulation, when I introduced today's lecture. So there's need to regulate gene expression because of developmental reason. For example, you are growing from single cell to two cells, then so mitotically you keep growing, and there comes a stage where these cells, they become different from each other and attain different identity. And this is only possible when we selectively switch on and switch off genes, which we refer to as differential gene expression. So that is developmental reason. Okay, cell identity, cell fates are established due to gene regulation, cell type specific gene expression programs. Then, of course, besides these developmental reasons, there could be environmental reasons as well. Whether you are prokaryote or eukaryote, if you are in a nutrient-rich, stress-free environment, there will be genes different genes expressed, and if the organism, be it bacteria or eukaryotic cell, you are under a very stressful environment, biotic stress or abiotic stress, there will be specific genes which will be switched on. Uh, Similarly, in case of pathogens, if you have infections of viruses, bacteria, etc., you know, organism fights back these intruders, which are bacteria or viruses, and there are different genes being expressed, switched on. We learned in earlier lectures that the bacteria is infected by the bacteriophages. So when phage comes in and infects bacteria, um, phage tries to hijack bacterial machinery, and in response, bacteria fights back the viral uh, invasion, and different set of genes or cascade is activated. If we talked about Nobel for chemistry in 2020, uh, the CRISPR system is basically a bacterial defense system against the viruses. So there are different reasons, and these reasons can be many, for regulating gene expression or switching on or turning off genes in bacteria or in living cells, bacteria or higher eukaryotes. There can be different levels of regulation. One level of regulation is simply transcriptional regulation. And transcription regulation can be like this. RNA pol2 may not bind because there are certain other molecules bound which exclude or which inhibit the binding of RNA pol2 to promoter sequences. When we say regulation of gene expression at transcriptional level, it means messenger RNA is not being formed. Transcription is not taking place, whether you are a prokaryotic cell or you are a eukaryotic cell. And when transcription is not taking place, we call gene is silent or gene is off. How? Transcriptionally off. Its transcription is not taking place. There are other levels as well. The other level is post-transcriptional level. In case of post-transcriptional level, let's say transcript is being made. In that case, transcription is taking place, but this transcript, which is RNA, messenger RNA, is getting degraded. This level of regulation is called post-transcriptional regulation. And third level is translational regulation, and translational level is where you have messenger RNA is intact, protein is either being made. If it is being made, it's quickly degraded, or it is inhibited by certain molecules, so protein remains intact, but it's inhibited from performing its function. It could be chemically modified. There could be some other inhibitory proteins which come and regulate this. It can be phosphorylated, whatever. So these are different levels of regulation. Today, what we are going to talk about is discovery of transcriptional regulation in bacteria, which was made possible through a very efficient genetic analysis. You will see today your concepts of molecular biology and your genetic analysis will go hand in hand. And what we are going to learn today is actually regulation of lactose operon in bacteria. How lactose utilizing or metabolizing genes in bacteria are regulated. This was discovered through genetic analysis. Much later, we understood the molecular biology or the detailed molecular biology of lac operon. Normally, if we compare prokaryotic and eukaryotic gene expression, when I say gene expression, you should know something should come to your mind that Tariq is talking about, um, in today's context of lecture is like gene is being transcribed or not. Okay. So normally in prokaryote, the default state is that genes are mostly on. Okay. Default state, genes are mostly on. RNA pol 2 is bound. RNA pol 2 is bound by other proteins, which are referred to as activator proteins. And this arrow means transcript or messenger RNA is being made. In contrast to prokaryotes, the default state in eukaryotes is off. Eukaryotic genome, DNA, is packaged in nucleosomal forms. Nucleosome is basically H2A, H2B, two different dimers of H2A, H2B, and then a tetramer of H3 and H4 proteins. Collectively, they are called histone proteins. So this is one nucleosome. Histone octamer having 146 base pairs of DNA going in 2 cycles like this, then to the 2 cycle. And this is fundamental unit. One nucleosome is fundamental unit of eukaryotic chromatin. When genes are packaged, and they are, as compared to bacteria, so there are 20,000 genes, for example, in human cell. The default state is they are off. Only those genes are on, which are, let's say, in liver, you will have housekeeping genes. Housekeeping genes are constitutively expressed genes, which are always on because they are performing housekeeping function. They are required for metabolism, respiration, energy utilization, etc. But there are tissue-specific genes, which are, only tissue-specific will be on, all the other developmental genes will be off. Okay, so the default state is ground state is off. In case of bacteria, if some genes are not required, bacteria switch them off by specialized proteins called repressor proteins. Repressors, they bind to specific sequence, DNA sequence within the gene, which are called operator sequence. These operator sequences are recognized by these repressor proteins, and repressor proteins, they block or inhibit binding of RNA pol 2, or even if RNA pol 2 binds, it does not allow transcription. They repress RNA pol 2 and gene is turned off. Bacteria is very intelligent. It does not transcribe any gene which is not required. Okay. In case of eukaryotes, when the ground state is off, this off gene is switched on by RNA pol 2 together with some enhancer binding proteins, some transcription factors. A huge, complex at promoter is assembled, which then switches on specific gene. What we are going to talk today is this kind of switch in bacteria where, and this, as I told you, this is about lactose utilizing genes. So,
I have a question.
G,
So, how does the default on and off state affect the organisms on a larger scale? I mean, the time it takes, and the nutrients, and the energy it requires.
for example, bacteria, let's say, because we are going to talk about bacteria today. In case of bacteria, it's growing in an environment, where it has glucose available. And remember, glucose is the most preferred source, carbon source for bacteria to grow. Now, bacteria also has other genes like lactose, because It could be you don't have glucose available. Bacteria has evolved another set of genes because in the absence of glucose, otherwise it will die. But in the presence of glucose, it will switch off lactose. So lactose genes are permanently switched off. It does not want to spend energy on metabolizing, breaking down lactose into glucose and galactose, and then utilize glucose as carbon source. Similarly, there is a galactose utilizing genes, etc. So did I answer your question that this kind of gene regulation so is very important for efficient growth of bacteria? Clear?
Yes.
Yes,
Sir, I
sir.
have a
Thank
question
you.
as well. Sir, I have a question as well. Sir, I was just thinking about examples of genetic regulation. So if we talk about like chameleons, let's say that change their color according to their environment. So is that also an example of genetic regulation? And if so, is it possible for genetic regulation to happen that fast? मतलब वो, in a matter of minutes, color change कर ले.
Very good
So
question. Excellent question. We call that, the chameleon example you gave is, we call that phenotypic plasticity. There are many organisms like chameleon, there are larvae
octopus.
which feed on catkin flowers, okay? And what they do, they adapt the morphology or phenotype of their surrounding to hide or to evade the predators. And yes, that is linked to, eventually, that is linked to gene regulation. And some of the uh, examples or known examples of such phenotypic plasticity are very fast and very quick. So that is possible.
Okay, thanks.
So we call this as, genetic switches in bacteria. And such quick genetic switches, they are due to, I have made this slide to make things easier. For example, you have a gene which has binding site for an activator, but this activator is bound by, so this activator at its own, this activator which is responsible for activating this gene, at its own, it cannot activate gene, rather it requires another molecule, and this molecule can be a metabolite or it can be another protein as well. But let's say there is a metabolite, a sugar-like molecule, or an amino acid, like it's mentioned here, tryptophan, or a sugar-like molecule. If it is bound, then it can switch on gene. And here the role of this, so this molecule is called ligand. If it is tryptophan, sugar, metabolite, or whatever amino acid or sugar molecule. In this case, binding of ligand to the activator is having a positive effect on gene regulation or gene transcription. But such an interaction can have a negative impact as well. And in that case, let's say activator is bound by a ligand, and this ligand inhibits it from binding and activating the gene expression. So in this case, ligand is having a negative effect on gene regulation by this activator. Both of these examples, such switches, they exist. If you go and read about tryptophan, operon, you will find a different kind of gene regulation as compared to what we are going to see today. In order to achieve such genetic switches, as I just showed you on this slide, So there are two types of protein and DNA interactions which are required. One is indeed you need to have gene promoter. So this is the promoter. And on promoter, we have RNA polymerase binding is required. However, as I told you in bacteria, the promoter is also followed by another DNA sequence, which is called operator sequence. So the second kind of DNA protein interactions take place at operator, and this operator needs to be bound by proteins which are either activators or repressors. If repressor is bound, which is here, it will have negative regulation and there will be no transcription. Repressor, which means it is repressing, having negative effect on transcription. If activator is bound, activator operator interaction is taking place. Now you will have a positive regulation and transcription is taking place. Promoter and RNA pol 2 interaction is in both the cases, because RNA pol 2 recognize specific DNA sequences which you must have read in BIO216, your molecular biology. You know the Tata box sequences in the promoter element, -10 sequences, etc. RNA pol 2 goes with the help of sigma factor in bacteria and is recruited on promoter. Now, if it is having activator next to it, gene will be on, but if it is having a repressor bound to operator, transcription will be blocked. Now, thinking of these ligand and protein interactions, if the activator is bound by a specific ligand and that ligand removes activator, now you will have no more transcription. And here, if repressor is bound by ligand and that ligand removes, that ligand has a negative effect on the repressor, repressor moves away and now have a transcription. So that is, positive regulation and negative regulation. So here in this case, this is positive regulation, and here in this case, it's negative regulation. Is it clear so far?
So I have a question.
Ji?
Sir, the activators and repressors have specific sites for the ligands, or any kind of molecule can come attached to them and lead to some
Very
conformation?
good question. Very good question. So that's the next slide. These answer to your question is these activators or repressors, they have very, very specific DNA. No, not DNA. These are proteins. They have very, very specific amino acid sequences which recognize which provide a pocket for binding to the ligand. So these activators or repressors, they have two specific domains. One is indeed the DNA binding domain. Why do they need DNA binding domain? Because whether you are activator or repressor, you have to recognize this this DNA sequence which is in the operator sequence. So these activators or repressors, let's say I try to draw a protein sequence here. Now, these amino acid sequence from here to here in this protein, they are DNA binding domain. They can recognize specific sequence in the operator. At the same time, they have an allosteric site. Let's say amino acid sequences from here all the way till here, which will serve as binding site for this ligand. And we call this allosteric site because binding of ligand to these amino acids, this particular domain decides which function protein is going to do. For example, here, activator is available, its DNA binding domain is available, but it cannot bind because it is not bound to ligand. Now, as soon as ligand comes in, it binds here. Now, it induces a conformational change in the protein, binding of ligand to this allosteric site result in conformational change you can see in the protein, and that is must for binding of DNA binding domain in the protein to this DNA sequence. And now you will see positive effect. Same can happen on repressor. Let's say repressor is bound to DNA sequence. Now, in order to remove this repressor, when you will have ligand bound to the allosteric site, specific amino acid sequences in this protein, it induces a conformational switch, which is unfit to remain bound to the promoter sequence. Clear?
Yes, sir. I have another question though.
Yeah.
How do the ligands know that they have got to come and attach to this specific site? Like, is there any signal or what? And also, does the concentration of ligands have an effect?
Very good question. So, how, two questions. How ligand knows where to bind? And second is concentration effect. First, I give you answer about concentration effect. Let's say this ligand is being utilized by this activator or repressor, because this activator or repressor is being transcribed and translated somewhere else within the cell, and you are providing this ligand, small, it is bound, this one is bound, this one is bound, okay? And if this ligand is getting metabolized, then there will be time when you will see, concentration of ligand is becoming less and less. As soon as the ligand is totally utilized, you will observe effect on the transcription or the gene regulation. Answer to your second question is how ligand know where to bind. That is mechanistic question. That's I think it requires maybe another 20 lectures of molecular cell biology to explain you how ligand. First thing is, of course, let's stay with bacteria. You add a ligand in the media, so the media is present, ligand is present in the media. And if bacteria requires this, because bacteria is under stress or starvation, it needs to bring in, then there are specialized proteins which bring in, then metabolize, then pass on to, let's say, these proteins. So there is a whole molecular pathway or mechanisms which provides this ligand to specific binding site. In eukaryotes, for example, in eukaryotes, so you have, this is cell membrane, plasma membrane, you have the nucleus. This one is the nucleus. Now, there are some proteins which are present on the cell surface, and ligand is coming from neighboring cell. And these proteins lying on the cell surface, they are bound by this ligand. And then this activates a cascade of reaction within the cell, which then have an impact on the chromosomes or gene regulation. Now, how this ligand finds this, what we call, this is called receptor, or this ligand moving into this cell and interacting with some protein here instead of binding there. These are questions, I would say $64 million questions, but there are mechanisms known in some case, but I would not say we are 100% sure about the complete detailed mechanism about how it finds. Okay. Did I answer your questions?
Yes, sir. And also, I just wanted to ask another thing. Sir, are the effectors and the repressors are specific for the ligands, right? But are the ligands specific for certain kind of receptors and effectors only, or can like one ligand have effect, like a similar effect on two different types of effectors or something like that?
No, they are very, very specific. They are very, very specific. If a
So,
ligand can affect two proteins, two receptors, it will be specific for those two receptors. It will not be going and switching on 10 other receptors non-specific. It's very
sir,
high level of specificity.
when we talk about two different organisms, then does the effect remain the same?
Yeah. So let's say it's experimentally proven. Sometime, you know, you can take a human gene and place it um, in mouse, for example, and it performs the same function, and vice versa. So this is
Sir,
evolutionary
thank,
concept.
thank you, sir.
Sir, I
Okay.
also have a very small question.
Yeah.
सर आई जस्ट वांट टू आस्क कि जो लिगेंड की concentration है can that result in the variable expressivity and the penitence that we learned about?
Yeah. Yeah. Absolutely. I'm going to explain this on next slide. So I told you we are going to talk about lac operon, which was, you know, one of the beautiful examples in terms of gene regulation, which served as, you know, often people say, what is use of, you know, studying bacteria? What is use of investing money in, you know, yeast or C. elegans? You know, these are useless organisms. I strongly, you know, dislike such a statement, because that is not what we have learned in last 100 years or so. In bacteria, Jacob and Monod, they learned that metabolism of lactose involves regulation of lac operon, and lac operon in bacteria exists like, you know, there's promoter, then there's operator sequence, and then you have, you know, in bacteria genes exist in like single promoter activate, let's say, a polycistronic messenger RNA. So coding sequence for gene A, gene B, gene C, all of them are one transcript. And then protein will be made A, B protein from same messenger RNA, but different reading frames. So same promoter regulate a gene unit comprising of multiple genes. We call this in bacteria operon. Okay. Now, what they why lactose or lac operon is important, or why bacteria needs this. So lactose is alternate, as I told you, glucose source, and not glucose, the carbon source. Here, if you see, lactose is broken down by an enzyme in bacteria, which is called beta galactosidase. So lactose is broken into or metabolized into galactose and glucose, and bacteria prefers to use glucose for its growth. Now in bacteria, the lactose operon comprises of genes. One is, I told you, beta galactosidase, another one is permease. So there are three genes. So A is, let's say, LAC-Z, beta galactosidase is LAC-Z, then the permease, which is LAC-Y. Permease is the gene which actually, as the word tell you, it is transporter. It transports lactose into the bacteria, and then LAC-Z breaks down lactose into glucose and galactose. ZY, and there's another gene A, three different genes in bacteria. Now, they were understanding this utilization of lactose by bacteria, and it was known that, you know, bacteria grows happily in the absence of lactose. You see the growth of bacteria, the cell mass or number of bacteria is exponentially growing. But the lac operon or utilization of lactose only takes place when you add lactose in the media. Now you see activation of LAC-Z and LAC-Y. Before this, LAC-Z and LAC-Y, or the whole lac operon, is existing in the off state. Now imagine the question two of you were asking about concentration. This is the lactose, okay? It's being broken down into galactose and glucose, and this is being utilized by bacteria to grow. If you add here, let's say X gram or X milligram, or let's say 1 millimolar concentration of lactose, with the growth of bacteria, will it remain here still 1 millimolar or less?
So I'm sorry, your question cut off. My internet had some issues. Could you
If,
repeat the question?
if at this point you add X amount of lactose and bacteria starts utilizing this, metabolizing this, and this is how the bacteria is growing like this, the mass of bacteria is multiplying. You think at this particular point, the concentration of lactose will remain the same, or it will be less or more?
So it should be less
Less.
because
So the answer to your question about concentration of ligand, so it will be impacted. There will be a time here, for example, maybe, where lactose will be 0. Clear? And when lactose will be 0, lac operon will go back to off state after some time. So in this particular example, lactose is, we know it, that lactose is a substrate, and we will learn that lactose is also ligand, acts as ligand as well. Okay. So as I explained you, the 3 genes, beta galactosidase, permease, and I forgot what A stands for. Najma, can you Google what A stands for? So what was already known, that it's a very rapid on-off system. Now, in order to understand lac operon, and this is concentration dependent as well. We just saw there can be again off state or of lac operon. There was a discovery of synthetic inducer, and that synthetic inducer is called IPTG, isopropyl beta D thiogalactoside. Now the beauty of IPTG is it is not broken down. It acts as a ligand. It behaves as a lactose molecule, okay, but it is not metabolized. Look, it acts as an inducer because lac operon is induced by lactose. So IPTG serves the same purpose. It's an inducer, but by itself, unlike LAC-Z, it's not substrate for LAC-Z enzymes. So using IPTG, you can switch on lac operon, and you can study the whole lac operon without worrying about such concentration dependent that it goes off and genes will be switched off again. They, through their genetic analysis, the biggest discovery for Jakob monos was discovery of LAC-I mutants. I think it's 50 years or more. maybe 50 years ago. They discovered a mutant which was constitutively expressing lac operon. How can we learn lac Z is on or lac operon is on? Let's say we have in the wild type case, wild type bacteria, normal bacteria. You add here, you show lactose plus and lactose minus. Okay. Sorry. There should be minus, there should be plus. So in the absence of lactose and in the presence of lactose, or let's say in the absence of IPTG and in the presence of IPTG, the ligand, we have lac Z gene, we have lac Y gene in the absence, in the presence. In case of wild type, in the absence of lactose or IPTG, lac Z will be on or off?
Off, sir.
Off. Because we can add substrate for lac Z, we can add substrate for lac Y, and we will see that Z is off. It's only on when there's ligand, and same will be the case here. Is it clear so far? In case of wild type.
Yes, sir.
Sir, before you move on, I have a small question.
Yeah.
Sir, जब lactose जो है, if it acts as an inducer as well as a substrate, so it acts as a ligand for the enzyme.
Yeah.
Then how can
which
it also
enzyme? Which enzyme?
For the lac Z का जो enzyme.
No. It's not ligand for lac Z. We don't know yet it's ligand for what. All we know, lactose switches on this operon, and lactose is substrate for lac Z. Lac Z breaks down lactose into galactose and glucose. We don't know what lac Z binds to. What is the receptor or molecule where, let's say, lactose binds? This is, let's say, lactose. This is the protein or DNA. We don't know where it binds, but we know lactose is substrate for lac Z. Clear?
Yes, sir.
Okay. So in case of wild type, lac Z will only be on if lactose is present in the media, and we can see lac Z is on or off by simply measuring the concentration of lactose in the media, because lac Z will be utilizing this. Clear? And we can say if concentration is becoming less, lac Z is on. Clear?
Yes, sir.
And lac Z is on, that indirectly tells us lac Y is on, because lac Y is going to provide lactose to lac Z. It's involved in transport. Now, they discovered a genetic mutant which they named lac I mutants. What was the phenotype of lac I? Phenotype of lac I was these mutants, they always expressed beta-galactosidase at very high level, which means in these mutants, there is no need for lactose. Even in the absence of lactose, these mutants are expressing high level of beta-galactosidase. And this led to hypothesis that lac I mutants, they have lost some regulatory component because repression is gone. What we know, in the absence of lactose, lac operon is always off. Is it clear?
Yes, sir.
Yes, sir.
Now something must have happened that even in the absence of lactose, the lac operon is permanently on.
Sir, I have a question.
Yeah.
Is it possible that instead of even in the presence of lactose, there's some kind of mutation that kind of does not allow LAC1I to be expressed? Sorry, LAC I.
Can you repeat your question, please?
So, in this mutation, beta galactosidase is always expressed. Is it possible that even in the presence of lactose, beta galactosidase will never be expressed? Like, can some mutation result in that as well?
Yes. Yeah. We are going to see them.
Okay.
So let's take each mutant one by one, because they discovered what you are telling. They discovered all these mutants: lac Y minus, lac Z minus, lac I minus. So we are going to learn one by one. So let's now solve problem of lac I. What it means? Hypothesis is some regulatory component is mutated, which is required to keep lac Z in the off state. Okay. Let's repeat what we learned last time in the last lecture about, you can see what is here, what this word is telling you.
So F as in the fertility factor.
What it is showing?
So fertility factor, right?
It's F prime was not the fertility factor. That was F factor. F prime was used for a very specialized state of, in a very specialized experiment. You remember?
Bacterial conjugation.
These F prime plasmids, you remember, we isolated from HFI strains where, bacterial chromosomal markers were passed on. So you see now we are going to use F prime plasmids here to generate merodiploid and do genetic analysis in bacteria. Now, do you understand why in previous lectures it was important to learn F factor and F prime plasmids? Because eventually in genetic analysis, we are going to use them. So now let's pay attention and sort out the enigma of lac I mutant. So they said in lac I mutant, the beta galactosidase is constitutive. Even in the absence of IPTG, in the absence of lactose, beta galactosidase was constitutively active, always on. You add IPTG or lactose, regardless of presence or absence, it's on. So your interpretation is that I minus mutant is expressing beta galactosidase constitutively, always on. In the wild type state where you had I plus or Z plus, I plus Z plus, this is the wild type. It's not constitutive, it's inducible. Leg Z is inducible. Okay. Now, to determine whether I minus is phenotype is recessive or dominant, you generate merodiploid. In your mutant I minus Z plus, you transform F prime plasmid and F prime plasmid with wild type copies, because you want to see whether it's dominant or recessive. And it becomes like wild type. In the absence of IPTG, it's no more constitutive. Only when you add IPTG, this becomes active, utilize lactose. And we conclude that I minus phenotype, which was this one, where Z was constitutive, I minus phenotype is recessive. Is it clear?
Yes.
Okay. Now they said, okay, they had a mutant which is Z minus, mutation in beta galactosidase. If you have I plus and Z minus in the same organism, it's uninducible because Z is mutant. When beta galactosidase is mutant, there's no one who is going to utilize lactose. You will not see any effect on the lactose concentration, which means lag Z is, when it is mutant, it's uninducible even if you have wild type copy of I. You determine beta galactosidase mutant is recessive or dominant. You generate merodiploid state, bringing in wild type I plus Z plus. And what you discover now, when I add IPTG, Z is inducible, which means this phenotype of Z, which was uninducible, is recessive to wild type. Next one is very important.
So I have a question.
G.
So if we have like mutants that are like a lack Y minus and it has the ability to synthesize beta galactosidase, but if we have like I gene which is still intact, then what will happen to beta galactosidase? Like if we add lactose to the medium.
Which mutation we have now?
So lack Y minus.
उस पे आते हैं अभी, उस पे अभी आते हैं. Let's go one by one. पहले let's sort out ये I और Z का relationship क्या है, ये क्या चीजें, ठीक है? Now they said, okay, let's find whether they complement each other or not. In the mutant, they have lag Z mutation, wild type, but on F prime plasmid, they have I minus and Z plus. They found that this is inducible, just like the wild type, it's behaving as wild type. And conclusion is, this is complementing this one and this one is complementing this one. It means I minus and Z minus, they are in different genes. Is it clear? So this concluded that gene which gives I phenotype and gene which gives Z minus phenotype, they are actually in two different loci or genes. Because why they have to show this? Because I phenotype is being read by constitutive activity of beta galactosidase. So this clearly told them that complementation is taking place, because this is on bacterial chromosome, this is on plasmid, two totally different DNA molecules, and they complement each other, which conclude that they are two different genes. Is it clear so far?
Yes, sir.
Yes, sir.
Okay. Now, what is I and what is Z? Z, I already told you what it is. They also determined whether they are recessive, dominant. They also, through complementation, showed that, they are in two different genes. Through these genetic mutants, through genetic screen, they found many different mutations which affected the structural genes of lac operon, which are lag Z, Y, A, etc. And we just analyzed lag Z, and the other one was lag I. They also did analysis whether these different mutants they discovered, they are dominant or recessive using F prime plasmid. And their conclusion was that, already they knew I and Z are in two different genes. This slide actually should not have been here. This should have been in the end. This should have been earlier. Okay. So what they concluded, that their conclusion of their genetic screen result turned out that, the repressor, which is I, was named as repressor. Why? Because in the absence of leg I, Z was constitutive, always on. It means I, which is a totally different gene, is blocking expression of leg Z in normal wild type case. If we mutate leg I, which means leg minus, I minus, now leg Z was constitutive on. It means in wild type case where I plus is there, it's acting as repressor. Having this slide here disrupted my order, the way I developed this lecture. I don't know why this screen and this slide came in here. So we are going to see the operator sequence, relationship of I and Z I just explained you. They assume this as a repressor, and lactose was already known to be substrate of Z, and this was being used as a readout for phenotype of leg I. So in other mutants, what they discovered when they found so many other mutants, O plus, Z plus, Y plus, these are genotypes. If you have, and this is wild type indeed, plus means wild type. In the absence of in the absence of lactose or IPTG, which means non-induced, the beta galactosidase, this minus plus is the readout. Under uninduced state, in the absence of lactose, leg Z is off. In the presence of lactose or IPTG, leg Z is on. In wild type, under uninduced conditions, leg Y is off, and under induced condition, leg Y is on. And we call wild type is inducible. That is what we knew already, that only under induction or only after adding lactose, this operon gets active. Now, pay attention here. They have everything wild type, but they brought in on F prime plasmid where O is wild type, Y is wild type, but Z is mutant. And they already knew that this one is recessive to wild type. So they showed that under uninduced condition, it remained uninduced. It only got induced when you have inducer there, which means Z plus is dominant over Z minus, and there was no effect on induction. So it pretty much behaved as wild type. They found a genetic mutant which they call OC, or constitutive operator. OC was constitutive, they named it constitutive operator. And in this case, the phenotype of OC, why they named it OC? Because whether you have lactose or you don't have lactose, these genes are on in the presence of this mutant. You try to determine whether operator sequence OC, it acts as, dominant, recessive, how it behaves. Look, you bring in wild type. In this bacteria, this is not wild type. Pay attention. Wild type operator, beta galactosidase is minus, and wild type permease. In F prime plasmid, you brought in constitutive operator together with wild type beta galactosidase and mutant permease. If I draw here, sorry, if I draw here, what you are doing in bacteria, let's say this is the bacterial chromosome. You have O plus, Z minus, and Y plus. And on the F prime plasmid, let's say this is F prime plasmid, you have O constitutive, Z mutant, sorry, Z plus, and permease minus. Now, what they discovered that beta galactosidase is constitutively active, which is just like what they observed here. But the permease is no more constitutive, it is inducible, which means this OC, this constitutive operator, is only acting on or effective for the genes which are next to it on the same piece of DNA. Same piece of DNA. OC, Z plus, Y minus. And which is other piece of DNA is the chromosomal DNA. The chromosomal DNA is what? O plus, Z minus, and Y plus. So this constitutive is only helping this one. It's not helping this one to be constitutive. This one will only be on when lactose is added in the media, or IPTG is, this operator, wild type operator, will become induced, or the gene will, and Y will be on, which is this, inducible. The conclusion is that operator sequence is actually cis acting. Cis means along the same length, along the, if this is, my arm is, lac operon. If operator is on this piece of DNA, then any gene lying next to it, Z or Y, provided they are wild type, they will be on. But this constitutive operator, it cannot help DNA on this arm, which is not the same molecule, not along the same arm. It's on different. So this one is called cis acting or cis regulation. So operator is a cis regulatory element. Just like you may have read in molecular biology, promoter element of a gene is cis regulatory element, because this promoter activates gene, only a particular gene which is lying next to it. A promoter of a gene cannot activate, a totally other gene who has his own promoter as well. So this was the definition of cis acting, but this was discovery about cis action of operator. Is it clear so far?
Sir, just to clarify,
Yeah.
ये जो mutant O है, basically ये करिए रहा है कि जो Z positive, जो Z plus है, जो wild type Z है, उसको basically मतलब it's not allowing it to activate, right? Because it's mutant itself.
No, it is allowed it to activate. Constitutive operator means, look, Z is constitutively active, beta galactosidase. You don't need to add lactose for metabolism of, clear?
Yes, sir. Yes, sir.
ये प्लस जो है इसका मतलब बीटा galactosidase is on. Minus means it's off. This is telling that this OC, this one यहाँ पे, which was, let's say, F prime plasmid, this one, this OC is regulating only expression of this one, this structural gene, but it cannot activate something on different chromosome. Did I make it clear?
Yes, sir.
Sir, but why? Sir, I don't understand. Like, why can't it activate some other gene on some other strand of chromosome?
So answer to your question will be on next slide. Just hold on for.
Okay.
Okay. So this was very important. This is very important. Operator is cis acting. Now, what we have learned so far? We have learned Z, Y, A, these are the structural genes of lac operon. We have an operator sequence which only acts to its own regions. Okay. And what we also learned that, you know, the lac I was a different gene and lac I was a repressor molecule, because in the absence of lac I, Z was constitutively active. So we have now learned things. Lac I is a repressor actually, which keeps Y and Z completely off in wild type state. If we get mutation in lac I, which is I minus mutation, Z and Y become constitutively active. Also, if we have operator mutation, OC, Z and Y become constitutively active. But important lesson was operator only acts in cis, it cannot act in trans. Opposite of cis is trans, that on other chromosome also it can go and act. Now, what turned out that when in the presence of constitutive operator OC, when lac Z and lac Y, they both are constitutively active, hypothesis emerges that, you know, repressor, which is I, is somehow in wild type case binds and represses transcription. So in case of constitutive operator, this repression is not taking place. It means I, even though it's wild type, it's not able to bind the operator. Okay. So in order to resolve and understand that repressor I is actually a molecule which binds and then represses, they again combined. So this is the wild type I plus, so wild type repressor Z and Y, and we know in wild type Z and Y are inducible. They are only inducible when you add lactose or IPTG. So we call I plus as inducible. Clear? So normally what happens, I, let's say this is the lac operon, and lac I is somehow binding, and when we, and now it represses, so Z and Y are off in wild type. If we have added lactose or IPTG, this repression is no more there, because we see in the presence of IPTG or lactose, Z is turned on and Y is turned on, which means this lac I repression is inducible. So lac I is inducible. It's removed. Its repression caused by lac I is removed, and Z and Y are turned on. ये तो क्लियर है ना यहां तक कि मैं बार बार कर रहा हूं ताकि ये लैक ऑपरेंट वाइल्ड टाइप आपको समझ होना चाहिए. If, and we already knew in the LAC-I mutant, they are both constitutively active. This next experiment is important. They had wild type repressor, beta galactosidase mutant, and Y plus. And they brought in F prime plasmid in this mutant, in which you have I minus mutation, wild type for Z and plus. What should be the phenotypes? You should, so based on the phenotype, there will be a big conclusion. The phenotype turned out when you had I minus mutant, I minus, Z plus and Y plus, they both are still inducible. It means I plus is dominant. I plus is dominant over I minus. In the absence of, if you remember, LAC I minus, Z and Y were both constitutively active. You remember this one? In the absence of LAC I, they're both constitutively active, but they become inducible in the presence of wild type, which means I plus is dominant over I minus. Final mutant analysis is even more interesting. If you have I minus, Z minus, and Y plus, but you have normal repressor and Z plus and I minus, what you expect? You expect in the absence of this I minus, it should activate this Y plus. But it is not the case. It still remains inducible, just like here. And you conclude that I plus is actually a trans-acting factor. Trans-acting.
Sir, I'm a little confused about this part.
Yeah, I'm explaining. I'm trying to explain you now. So let's say you have I plus. Here it was also I plus. Okay. If we draw this one, it will be more easy here. I minus, Z minus, Y plus. What should be genotype of Y plus and Z minus in this bacteria? Only this one.
Sir, Z should be non-inducible.
Yeah, Z is mutant, non-inducible, minus minus. What will be Y plus?
So it should be inducible.
Always.
It will be inducible or always on?
Always on.
Always on.
Always on. Because it's LAC I. In the absence of LAC I, you remember this was the key repressor. In the absence of LAC I, Y will be constitutively on. Now, if in this bacteria you brought in F prime plasmid, where you have wild type repressor, you have Z plus, and you have Y minus. All this is in one bacteria. Now tell me phenotype for beta galactosidase and phenotype for Y plus in this bacteria where you added F prime plasmid.
So for Z, it should be inducible because the repressor is on.
Z is inducible, okay. And what is for Y?
So it should also be inducible because the repressor is on.
Why?
the repressor is present.
Here it is I minus. It should be causing no repression on this locus. So it should be constitutive, no? What do you think?
Sir, but on the complementary gene, there is a repressor, and since that is dominant over the I plus is dominant over I minus, so that's why the repressor will still be produced, and Y will not be activated.
Anyone else? Who else would like to try? So what I think Rafe was talking.
Yes, sir.
So your understanding is right because it's dominant. So Y plus should be inducible. Others, I would like to hear from others as well.
Yes, sir, I agree. Why it should be inducible because the dominant will overrule the recessive trait.
But
of
there's
I
a
minus.
very good. But there's a bigger conclusion being drawn here. The bigger conclusion is I is a trans-acting factor. You remember we learned from operator, it is cis-acting. Operator, when it is mutant constitutive, it is only acting on genes on the same DNA molecule. But repressor, it's a factor. It's not a DNA sequence. It's a repressor. So repressor, if it is present even on this DNA molecule, it's being transcribed, translated, okay? And it is able to repress DNA here and DNA here. That's why this Y turns out to be inducible. That's why it is dominant to this mutation. That's why I plus is dominant to this, because once you have wild type, its protein is being expressed and it is going and binding to its target sequence in the operator region. Operator is the region where repressor binds. If you have I minus and I plus, this is enough. This is dominant over this one. Okay. Even though this is I minus and you say, oh, all of them should be on. Let's say this is wild type, hypothetically. Okay. It will not be on because this repressor being produced from this molecule, this chromosome, can go and act on this operator, keep them under control as off state, and they will be only switched on. They will be inducible only in the presence of inducer. So we call this trans-acting factor. This is no more cis. This is trans-acting. Operator is a cis-acting molecule. Clear?
Yes, sir.
So I'm still a bit confused about this, about how it's about the transacting operator.
So let's see on this figure. ये आ गया इधर आपका question का answer. You have a heterozygous state, a merodiploid. Is it a merodiploid? This one. This is, let's say, bacterial chromosome, and this is your F prime plasmid. You have mutant repressor and wild type locus. You have on the F prime plasmid wild type everything. In the absence of lac repressor, this should be on, permanently on hona chahiye na? Is it true, Rafe?
Yes, sir.
But it does not. यही है ना वो जो अगले पेज पे हम एक्सप्लेन करें वो ये वाला. They are inducible. Why? Because repressor molecule being transcribed and translated here, this is the protein I. It goes and binds to the operator sequence on both, sits there and blocks transcription. Transcription is switched off. Z and Y will only be on when this, let's say this is your repressor is sitting here. ये वाली protein इधर जाके बैठ गई. You added lactose, which comes, binds to this repressor, takes it away, and now Z is on, Y is on. We call this transacting because molecule being produced, transcribed. So the wild type gene is only here on the plasmid. The gene being produced somewhere else in the genome, its product is going and acting altogether on a different position. Had this molecule restricted only and only to its own chromosome, to its own, then what is it? Cis-acting. जो कि operator है. Operator and promoter are cis-acting molecules. If you have promoter, let's say you have a promoter minus, what will be the effect on Z and Y? Promoter minus.
So it won't be produced because
They will be?
they won't be activated.
Very good. Very good. So they will be off, permanently off. But this promoter minus mutation will not switch on these wild type genes on other molecule, the F prime plasmid. And due to this effect, we call promoter and operator cis-acting elements. But repressor is trans-acting because you are providing on F prime plasmid. Protein is being made, produced from only this wild type locus. This is going to the wild type operators, wherever operator is present on bacterial chromosome, on F prime plasmid, and switching them off. And they are inducible. Did I made it clear?
Yes, sir. I was just confused because I thought कि जो trans होता है वो Z की जगह Y पे act करेगा.
No. So is it clear so far?
Yes, sir.
So the
Yes,
last
sir.
slide is they
Sir, I
found.
have a question.
Yeah, please.
Sir, we did talk about promoters, के वो cis acting होते हैं, trans acting होते हैं. Sir,
Yeah.
enhancers can be trans acting or cis acting.
Enhancers can be trans-acting as well. They can be far away somewhere else and acting on multiple genes as well sometimes.
And sir, do they have effects on like transcription or regulation of genes?
Of course, enhancers होते ही transcription regulation पे, वो हम जब eukaryotes में आएंगे उधर बात करेंगे. So now they also discovered genetic evidence for allosteric. You remember we talked about allosteric sites in repressor, etc. So they discovered another mutation which they named as super repressor. Super repressor. You have lactose, you don't have lactose, you induce, you don't induce. In the presence of super repressor, IS mutation, everything is off. Phenotype. Phenotype was, they discovered a mutant phenotype in which beta-galactosidase and permease both were permanently off, always, where you add lactose or IPTG, whatever. And when they mapped the mutation, they found that mutation is in the repressor region. They named it super repressor. Now, when they try to complement and find dominant and recessive relationship of super repressor with wild type, look, I plus Z plus Y plus I super repressor Z. The guy remains off always, and the conclusion is 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 super repressor is dominant over I plus. It means IS molecule, whatever the protein is, it's not responding to your IPTG induction or lactose. molecule. And it was later on discovered that the repressor is actually, so this is your total lac operon, lac repressor. It gets transcribed, repressor protein is being made. Repressor protein comes and bind here. So repressor is normally a homotetramer. Okay, 4 different molecules of the same protein is bound to this DNA sequence, which is operator. You bring in lactose, you add, this is the inducer, which is lactose or IPTG. It comes and bind to this allosteric binding site, ligand binding site, and it removes away repressor from the operator, and RNA polymerase starts transcribing. And lag Z, it becomes now lag Z plus, lag Y plus. In the absence of inducer, it remains bound and genes are off. Is it clear so far?
Yes, sir.
This is just this is same explanation of the previous slide. So RNA polymerase, it transcribes the repressor. Repressor protein gets folded. You can see this is its allosteric binding site, this one. It repressor binds to the operator sequence and keeps them off. Everything is off. You bring in lactose in the medium, it binds to the allosteric binding site here and removes, keeps it away from the operator binding site, and now RNA polymerase, which is bound on the promoter, switches on. Messenger RNA is being made. This messenger RNA, it contains reading frame for beta-galactosidase, permease, and I think, Najman, did you look at A? Why I'm forgetting about what A? Yeah. A. Come on. Is Najma there? I think her net is not working today. So all 3 proteins are translated from this. Yeah, I did not want to say acetyl transferase, but it is trans-acetylase. So this A. So proteins are being made, and then you know, okay, Z is inducible, Y is inducible, A, etc. is inducible. What after advent of recombinant DNA technology and molecular biology, we discovered that OC mutation, which we named constitutive operator, where in the presence of OC, you remember beta-galactosidase and permease both were constitutively active. And that's why we named this phenotype constitutive operator. We have learned after sequencing the DNA of this constitutive operator mutant that the binding site of repressor, so operator sequence is 17 to 25 base pair DNA sequence just before the lag Z, and there are specific mutations here, 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 here. So for example, GC wild type sequence is converted to TA, or this GC converted to TA, and this mutation has disabled binding of lac repressor I. Protein which recognize wild type sequence, this one, and represses transcription, this repressor binding site, DNA sequence for repressor is mutagenized, and now lag Z and lag I both are constitutively active. I think you will appreciate the power of genetic analysis, how they, solve molecular regulation of lactose operon purely through genetic analysis, through these genetic mutants. We only learned that this operator sequence is actually mutated, the binding sites for repressor is mutated only once we learn, sequencing of DNA, which is in early 80s or so. We found that this one is indeed a transacting factor. It encodes a protein, it represses only once we had molecular tools of recombinant DNA technology in our hand. Whatever they discovered through genetic analysis, using F prime plasmids, using making merodiploids, and creating genetic mutants through genetic screen, analyzing dominant, recessive, complementation tests, cis-acting, trans-acting, everything holds true till today. Molecular biology only proved them right. So with that, we are done with the microbial genetics module, and you have to tell us when to have the final the exam for this module. We still have quiz for this module next week, and I think take 1 week, max 2. I would say don't take more than 2 weeks because your concepts, I think 2 weeks are more than enough for your preparation. I would suggest you should come up with a date for next exam, not next week, but the week after. Okay? Any questions? If there are no more questions, I wish you all a good day. Okay?
Allah Hafiz, sir.
Allah
Allah
Hafiz.
Hafiz.
Purpose.